Dear participants, today we are having our guest Dr. Swathi Alagesan, who is a DST-Inspire faculty fellow in the Institute of Bioinformatics and Applied Biotechnology, Bengaluru. And she has completed her bachelor's in engineering from Delhi College of Engineering. Then from the Department of Chemical Engineering, IIT Mumbai, she obtained her PhD under the guidance of Professor Pramod Vangika. And she worked on studying the circadian rhythms of cyanobacteria. She visited University of Nottingham for her postdoctoral work to join with Nigel Minton's group in Synthetic Biology Research Center. And during that period, postdoctoral period, she was involved in generating synthetic biology parts and using them for metabolic engineering of autotrophic bacteria. 2017, she joined Anna University, Chennai as a DST-Inspire faculty, and then she moved to IBAB Bangalore in the month of August of 2020, that's recently, and her current research is focused on generating various synthetic biological tools and using the insights obtained from flux analysis to engineer microorganisms. So she is having a very strong background in synthetic biology, in developing the tools using prokaryotic systems. So on behalf of the organizing committee and authorities of Madurai Kamaraj University, I welcome you, madam, to deliver your valuable talk in this AICTA approved and AICTA sponsored ATAL-FDP program on synthetic biology. Now I handing over the session to Dr. Swathi. Swathi, please.
Thank you, sir. It is actually my pleasure to be able to present a bit of what I know. And I also encourage, if there are some feedback, please do give me so that I can improve myself. So my presentation is on synthetic biology tools for prokaryotic systems. So I will kind of give you a brief background, which I'm sure there have already been a lot of lectures on synthetic biology, so you have a sort of an idea about synthetic biology. I have a few slides. I'll begin with just giving you an overview of synthetic biology and then go into the various tools. And it is a combination of both what is noted in literature as well as a few insights that I have seen during my research as well. So to begin with, I am sure you all know what synthetic biology, but just if you split the term, then you have synthetic and biology. So we see a lot of synthetic materials in our day-to-day lives, be it synthetic fabric, synthetic polymers, or plastic, rubber. So when we think of the term synthetic, what exactly comes to our mind? It is anything which is made by chemical synthesis. So, and we want to imitate a natural product. So if there is a fiber, we are making it by chemical synthesis and we are trying to imitate it. And when it comes to biology, so what is synthetic biology? Is it making a synthetic DNA molecule, or is it making a synthetic cell, or is it building a whole genome through chemical synthesis and playing God? So there are different aspects of seeing, and the presentation will be more basic, where we are just looking at the DNA or gene level. So when we think of synthetic biology, there are a lot of definitions given. So there are mainly two aspects, where we are designing and fabricating a biological component or a system, and that does not already exist, or we are redesigning a system which is already existing in biological system. So how is synthetic biology then different from genetic engineering? So genetic engineering is more traditional and basic, whereas when we come to synthetic biology, we are looking at a more standardized system. So aspects like regulation, standardization, modularity, these are come into effect when we are thinking of a synthetic system, because we want it to function in a specific way which we can predict, and it does meet our expectations. So synthetic biology is not just concentrating on biology. We have other components like engineering, bioinformatics, mathematics, systems biology, computational. So knowledge from different aspects kind of contributes to synthetic biology, which is at the core of um, all these concepts. So there are main approaches which in the definition itself we have discussed. So there is a top-down approach where you are starting from a chassis and you are kind of engineering it to make it an optimum system. Whereas in a bottom-up approach, you're starting from synthetic nucleotides. You're building amino acids and you are making a whole device. So that is the bottom-up approach. And this is actually, if you can see my cursor, so this is like a timeline of how synthetic biology has evolved. And what I am going to deal in my presentation is mainly what has happened in the last decade. So developing parts, modules, and then using them for various applications. So my application is basically focused on bioenergy and production of platform chemicals. So coming to the design and process cycle. So whenever you think of not just biological system, you think of anything that you use, be it cars or any computer, anything. The development process involves a design process where you are designing it, then you are building the system, then it undergoes testing, and after testing you learn what the shortcomings are of a system, and then you go back to designing so that you can optimize and build a proper whatever cars to computers to whatever technology you're building that is suitable for your use. Even in synthetic biology you have your design process where you are kind of designing what you want your system to achieve. So it can be a knockout system, maybe you want an organism to behave in a specific way, or you want an organism to produce something. So that comes in the design process. Then it undergoes a building process where you have to assemble the system. You have your parts, tools that come in, and you are assembling and building a synthetic system. Then you have to test the system, whether it is performing to your expectations or whatever function you wanted to do. And based on the test, you will learn and go back to the design process where you will optimize it further. So my presentation basically is focused on first component, design and building. So tools for design and building, I discuss the parts library. For tools for testing, I will discuss about biosensors, and again, on tools for learning, I will kind of give you a brief overview of flux analysis. So these are just specific topics under these. This does not encompass everything under design, build, test, and learn, but just a few tools that are used for each of these steps. So starting with the part library. If you dismantle a car, each part or each piece is defined as which comes together to make up a whole of something. So there are around 30,000 parts in a car. And if you look at what are the key aspects of and just hide. So what are the key aspects or characteristic of a part? It is a result of a careful design process. It should function according to the design, and it should behave as you had predicted in the design. So these are the of any part, be a part of a car, part of a computer, or any technology. The same thing then applies to a biological system as well. So in a human body, there are parts. You go on breaking it down to the simplest part. You come to a gene. So I am sure everybody has heard about the central dogma, where DNA gene is transcribed into an RNA and then is what actually reflects in your physiology or characteristics. So you can target each of these processes at specific points using some parts, and these are the parts that we are focusing on here, and I will deal with each of the parts how you can tweak them to kind of get your synthetic system. So when prokaryotic, sorry, is there a question?
No, please carry on. Nothing.
Okay.
Yeah.
Okay. So there are different types of genetic parts. So in a prokaryotic system, which is more simpler genetic, you have a promoter system, you have a ribosome binding site, and your protein coding sequences and terminators. I'm assuming that basic molecular biology everybody is aware of, but just to briefly say, promoter is where your transcription begins. Ribosome binding site is more useful during translation for your translation initiation complex to form. Then your protein coding region is your protein of the gene of interest, and terminators where the transcription terminates. And eukaryotic systems have additional components which are like insulators, enhancers, or if they even the protein coding sequences is divided into exons and introns. So here we'll just concentrate on the prokaryotic. And again, all the 3 points while designing these parts is important. Is you have it should be a result of careful design. It should function according to the design, and the function should be what you have predicted design process. So if you haven't already seen, you should visit the iGEM website. There is a registry for standard biological parts. So you get a list of all the parts that are there. So what for imagination purpose, you can think of a car with its nuts, screw, bolts, and different types of parts. And you can think of a biological system which is composed of a promoter, RBS, protein coding region, terminators. What you want to do is you want to pick up the parts you want and you want to build a system. Just like you can assemble a computer or even a bike to your liking. Similarly, you can assemble a biological system as well using these parts. And it behaves in the way that you want it to behave. Coming to the promoter is like I've already mentioned, it's a DNA sequence that recruits transcriptional machinery. So here is the operator sequence. In case of there is a repressor protein, it will go and bind. So promoter, your RNA polymerase cannot move forward for transcription. However, in the presence of an inducer molecule, this is relieved and transcription can happen. This is an example of an inducible promoter system. And so there are different promoters. You have your constant promoters and your inducible promoters, which can be positively or negatively regulated. So I'm assuming everybody is familiar with the concept of promoters. So coming to synthetic promoters, now we know that genes are under the control or under the control of promoter sequence. But if you want a stronger promoter, you want a synthetic promoter system, how are they designed? So the designing process of promoter system is usually you have conserved sequence, the Tata box in the -35 region. So these regions are kept constant. The nucleotides between these regions are then through randomization, you can create different sequences, test them to make a library of promoters, and based on the strength, then you can choose it to develop your synthetic system. So these are the different methods: saturation mutagenesis, error PCR, and hybrid promoter, which is explained in this image. So you can keep the conserved regions constant and carry out mutations in these regions, which are marked as N. And then series of different sequences, you can clone them and test them and get your library of different synthetic promoters of varying strength. So another approach is hybrid promoter engineering. where different promoters which are naturally from see different genomes or you take you have this library, and then you can mix and match the different core promoter sequences, the activator sequences, or the upstream and match to get a combination of different hybrid promoters. Now you can test them, and based on the strength and your application, you can choose the promoter of your interest and use it for developing your synthetic system. And there are computational methods as well. So this is an example, a diagram showing how it can be done. So computationally, you can kind of train your software with a training set known strengths. Based on that, based on yeah, I think so. I can hear myself. So I think somebody's speaker is on.
Say echoing. Yeah, we are checking
Yes.
whether somebody's check. Deepika.
Okay. So based on the training set, you can actually develop a model or a consensus sequence here, and that can be used for predicting unknown sequences of what the strengths could be, and that can be tested, and this again goes back into training process. So a lot of computational tools are also available. Similarly, for RDS, site is important during translation initiation. So it determines the translation initiation rate and the stability of your mRNA. So you can keep the promoter constant. You can change your RBS to get a different expression of the downstream gene. But when you're looking at an RBS, you cannot just look just at the 5 to 12 base pairs here, because what is upstream and what is downstream also regulates the strength of your ribosome binding site. Design of RBS, there are few critical parameters that are kept in mind. One is RBS, the organism that you are going to use is also important, because the 16S rRNA sequence needs to be highly complementary to your RBS. The next is it should have minimal secondary structure forming, and there should be optimum spacing between the RBS and your start codon, and also the upstream bases, that is S1 ribosomal proteins will bind upstream. So that is important to RBS. So there are few online tools, few of them I have used personally as well. And if you look at all tools, they will ask you for upstream sequences. You are going to clone. If you see the algorithm calculation, it depends actually on the mRNA degradation rate, the spacing, how many nucleotides. So it takes into account both your upstream, downstream region also to determine the strength of your RBS. Now coming to terminators, which is again an important part, because you want the transcription to terminate and not carry forward, because you would have closed some other unit downstream. So a lot of times you want a proper terminator to be there in your synthetic constructs. So mainly we discuss about row independent terminators, which are more sequence dependent. So the structural part that is important in design of terminators is it should have the stem loop structure. With a poly A region, it can work as a bidirectional terminator, and this T region, the length of which is very important as well. So this it should form a proper stem loop structure with so the bases forming the loop is important. The base pairing here during of the stem is important, and the length of the T region is also critical. So now if we have all these parts, say we have the promoter, we have the ribosome, we have our gene of interest and the terminator. So these comprise our parts. Now we can form either with multiple parts like these, we can form a pathway, or we can just form one module. But we need to assemble them together. Now, how can we do it? We can either do it sequentially, say one at a time, you can clone it, or you can put everything together and kind of ligate it into one module, or you can think of libraries. So if you have a library of this, you can make all combinations that you want and choose the best system. That is combinatorial assembly. And that is mainly important when you have a library. You want the best system that is out there. So you can do combinatorial assembly and the best system here. About what are the assembly techniques. I'm sure you must have heard about bio breaks, Gibson, or just PCR overlapping. So this is just an overview of how you can assemble these parts together and make your synthetic system. Now coming to combinatorial, I have my library of promoters ready, RBS ready, my gene of interest is already there, and I have my terminators also here. Now I can do all sorts of permutation combinations to build up different gene modules, and I have a library of all these elements which are here. I can just do a one pot in one pot, put all these modules together, transform them into my bacteria, a full library of mutants. This I can screen through various techniques to get the system that I am most interested in. And so this is how a combinatorial library is generated, and you can choose your optimum system. This is again an example of how it works. It's more like a high throughput system. You have your parts, different parts, all different combinations combined. You can do a high throughput screening method and get your large scale producing module and a system of interest. So however, when you have such a big high throughput screening, when you have such a big library of different combinations, you want to screen them. And for screening, one important tool could be using a biosensor based screening. So that would be my next topic. But before going to that, I'll just briefly discuss part libraries that I have done during my postdoc. And here we actually around 25 promoters were tested in this study, and we also generated a library of RBS sequences. So we have this varying range of promoter strengths and varying ranges of RBSs. And what we did was we combined the strong promoter with a with different RBS strengths and cloned it with an isoprene synthesizing gene. So isoprene is a platform chemical of importance. So then we see that different RBSs with the strong promoter can give different range of isoprene production, and we find that it is not necessary that a strong RBS would produce more isoprene than a medium strength. So again, you get a range. Now we'll choose our optimum system, which is here, and go for large scale production. So this is just to show how you can use a library of different parts, make a combination, and make your synthetic system. Now coming to the testing. So if you saw the previous slides, there were just like 25 promoters and 30 RBSs. But usually when you are making a library of synthetic promoters and RBS, you have a huge number of different candidates. And when you build a combinatorial library, you have a lot of them. So for your testing, you could use a mass spectrometry or a similar device, but it will take you a lot of time. And if you want to get a more easier method or more straightforward high throughput method, then bio is one such tool that can be used. So like I said, if you have a combinatorial library, you have different combinations of the different parts. What you can do is you can use either a reporter system, which is like a fluorescent protein, or you can use a selector, which is like an antibiotic resistant marker, say. And if your organism is producing this GFP, it will have high fluorescence. You can do a fluorescent based assay to choose optimum systems, or you can, if yours has an antibiotic selection marker, you can put it in antibiotic. Only those organisms will survive, and you can choose your system. So this is how a biosensor can be used for as a for high throughput screening. So, like I've already mentioned, if you don't have a high throughput screening, then you can depend on analytical methods. But analytical methods are low throughput, and there is a limitation on the number of samples that you can process in a day, especially if you have a very big library of mutants to screen. So here biosensor based screening can help. Like if you have a biosensor which is like this, where a repressor is bound to your operator in the presence of whatever your ligand of interest or specific condition, it is activated to give maybe a reporter or a selection. And if this is a strain and you develop your combinatorial library, then you can easily screen them maybe by. So there are different types of biosensors that are there. One is the transcription factor based biosensor, which is similar to the previous animation that I showed you. So in the presence of your input molecule, the repression is not there. So you can produce a reporter gene, and based on the output, you can detect whatever is being produced or not. There are another type is based on two component biosensors. So in the sensor kinase, which in the presence and initiating activating transcription of your fluorescent reporter, or if there are two components like a ligand binding domain or a receptor domain with a DNA binding domain, you can actually alter this to bind to your compound of interest and make it into a biosensor which gets activated when your compound of interest or ligand of interest is present. And these are again different types of biosensors where they are actually split into two components. So that is slipped and only in the presence of your ligand they come together and are activated. Even in this split protein, so in the presence of your target, they come together. The ligand binding domains are also split, so they also come together and form an activating complex, which can cause control your reporter expression. So these are just few different types of biosensors which can be discussed. The based biosensors like those based on RNA like ribozymes and aptamers, those are have also been adapted for developing biosensors. But just a brief overview of the different types of biosensors that can be developed. And if you see literature, there are a lot of different biosensors which have been made for prokaryotic systems to detect different analytes ranging from amino acids to central carbon metabolites to carotenoids and terpenoids. So there are different types of biosensors that have been made, and they have been made either from existing regulatory molecules, like even arabinose based promoters have been modified. So based on these, sorry, different types of biosensors are available and can be used for as a high throughput screening tool. So presently my work is on developing a biosensor, and it's I work with both a hybrid biosensor as well as a transcription factor based biosensor, and that is for not just for throughput screening. I'm using that biosensor for metabolic engineering as well, where it can kind of feedback regulate and work as a switch for turning on and off of the system. Then next, once we have tested, now we have to learn. So we built our artificial system from the different arts and tools, and then we use biosensor as a tool for screening to get our optimum sensor, optimum construct we want to learn if it is working to our expectation. So that for that, an important tool is metabolic flux analysis. I will not go into, there are different methods again for flux analysis, like um, FBA and 13C based. So I will just give you a brief insight on 13C based flux analysis. That is something I do in my lab as well, and it is a good tool to learn how the system is behaving. So why do a flux analysis? Right, aim is we have a wild type organism, we want to engineer it for synthesis. This is what I want to do. So I have designed knockouts, knock-in strategies. I have used synthetic components and engineered it, and I have tested an optimum sensor. But we are not sure about what is happening actually and in the metabolism of the cell. So the in-depth knowledge of the underlying metabolism is also critical for designing of this system. So for that, flux analysis is very important before designing as well as after the testing phase, and to learn what is exactly happening. So I do 13C based metabolics, 13C steady state metabolic flux analysis. So you can do a transient labeling as well. So this is the most basic. What we do is when the organism is in steady state, when it is growing exponentially, the exponential growth phase is known to be its metabolic steady state. During that metabolic steady state comes a window where it is in isotopic steady state. So what is isotopic steady state is, say you give an organism 13C fructose, which is just labeled with 13C, the rest are all 12C. And this goes into the system and is incorporated into all the intracellular metabolites. There comes a time point where this labeling profile will not change. So if there are 13Cs, 3 will remain as 13C during the isotopic steady state. That is when I collect my samples, and to know, it will give me a snapshot of the metabolism during this exponential steady state condition. So now another critical aspect in designing these experiments is that, say you have just a substrate with one carbon, or all the carbons in your substrates are 13C labeled. In that case, either you do a transient labeling experiment. You cannot do a 13C experiment because under isotopic steady state, everything is 13C labeled. So there is no differential labeling in all the intermediate metabolites, and you're not getting any additional information. So if you have to use either a single carbon molecule as your substrate, or your substrate is uniformly labeled, you use a mixotrophic sort of condition where one is labeled, one is not. That under steady state conditions, you get a differential labeling pattern. And why this differential labeling is important is you can trace this label back to the respective substrates, carbons, and that is what helps you to calculate the fluxes in different pathways and know which has more carbon flux or which has less carbon flux, to help you to decide knocking off or knocking in sort of strategies for engineering. So what the experiment actually looks like, you can grow your culture, whatever bacteria, prokaryotic systems in 13C substrate, and during isotopic labeling, isotopic steady state, you take 2 samples. Why we take 2 samples is we want this to be a steady state. So we match the labeling profiles in the 2 time points to make sure that it's actually a steady state. Then we do take the proteins and hydrolyze them to get the amino acids, derive them through GCMS and get the mass spectra. So this is just how a chromatogram looks like. You have all the different amino acids that we got, And if you just look at one peak, say, of valine, then you have the different mass spectra. Actually, for a 12C, it should only be 144, whereas now we have 13C. So for every label that has been added, you will have a +1 in the mass. And what this actually tells you is this is your completely unlabeled base fragment of valine, which has 4 carbons. So for every 13C that is added, you will get an N+1. If 2 are labeled, you'll get an N+2, 3 N+3, and all 4 are 13C, then you get an N+4. And why this is important is this is what is the information called the mass isotope moment distribution that is used by softwares to calculate the flux distribution through the different pathways. So as an input, you have your labeling information, you have the label that is there in your substrate, and you have the pathways which give you the carbon atom transitions. So the output can actually be traced back to the input through these carbon atom transitions to give you the flux values. So there are tools which are available, and you can use them to get the flux estimates through different pathways. And what this eventually gives you is flux like these. So this is like a central carbon metabolism map, and the flux values under different conditions. So you actually look at specific pathways where your carbon flux is going and compare under different conditions and see is it increased, is it decreased, can you reroute it through specific pathways, and that information is the learning process which will help you for further designing the system. So this is how there are tools for, this is just a brief overview of a few tools which I could discuss today. So you have design, build, test cycle to get your optimum system, few tools which are used for each of these steps. So this was my presentation, and I would like to thank everyone for this opportunity, and also thank you for listening.